Welcome. The purpose of this video is to standardize the methods we use to quantify images in our attempts to assess the level of muscle damage that has occurred within any given section. So briefly, what we'll talk about is a little background on immunofluorescence, which is the primary method we use to identify muscle damage. We'll spend some time talking about image J, what it's good for, things, and then we'll do a sample analysis. So at the end of the day, the goal is to turn an image that looks something like this over here on the left into an image that looks something like this on the right. Importantly, so we get these areas traced, but we also get the numbers associated with that, which will then allow us to calculate the percentage of area. It will also give us some idea of the characteristics of the individual lesions. Okay, so let's move on. So immunofluorescence, basically immunofluorescence is using antibodies, that's where the immuno comes from, with fluorescent molecules bounded to them. So the antibodies are protein that's produced by various animals to protect themselves as part of the immune system. And so we've sort of co-opted these antibodies because they can be taught to bind to different things, or they can be made to bind to different things, and then we chemically modify them with fluorescent molecules so that we can actually visualize them. For the most part, these studies that we're going to talk about are also assessing antibodies, so we're looking at for the endogenous mouse IgG. As I mentioned earlier, these antibodies, the immunoglobulins, are proteins that are just floating around the extracellular solution, surveying, looking for the presence of an antigen, usually of a virus or a bacteria or some other pathogen. But in this case, we're just looking at the total amount of IgG. What's interesting, though, is that when a cell of myocyte, particular cardiac myocyte, is injured, it loses its membrane integrity, and then these proteins, which are usually in the extracellular solution, can enter the center of the cell. And when we section through them, we can visualize these proteins, these immunoantibodies, within the cell. So determining the area where intracellular IgG is present can give us a good idea of where muscle damage has happened. And that can be, in many cases, is one of our main functional readouts of how well this heart is protected by various therapies or other interventions. So most of these analyses will be done with image J. So image J is software that was developed by NIH specifically for the analysis of images. It's freely available and is present on multiple platforms, so you should be able to download it relatively easily from wherever you are. You just Google the image J and you'll find you'll get to the site. Alright, so now what we're going to do is we're going to go through a brief example. So I have sort of a fake image which will allow us to understand some of the capabilities of image J, and then we'll move on to a more practical example. Alright, so let's pull up this fake image. Let's see, here it is. Alright, so this image here is obviously very contrived, but it has some important parts. It has some really dark areas, it has some kind of light areas, and it has sort of this background, and it has a lot of little noise flecks in here. Now all of these are characteristics that we will see within our normal heart images that we observe. So as we approach this, we want to quantify these areas, and we want to quantify the entire area, but we don't want to include these individual little specks. So the key behind this is the ability to threshold this image. So thresholding is pretty straightforward. So you can find it up here, image, adjust, threshold, or you can use Shift-Command-T. And that will pull up a little menu which has some sliders on it which allow you to decide what we're gonna call positive and what we're gonna call negative. So the thresholding will turn your image into a true black and white. So there's either it's black or it's white. So if we move the slider all the way down, you can see only the very darkest images are able to show up. So these guys have an intensity that is super strong and will show up no matter what. But you'll notice we missed this area up here that we were initially trying to catch. So we can then increase this, and we continue. Oh, there it is. Okay, so now we're able to get all 3 of these images. But you also note we get some noise in here. Okay, so now what we want to do is we want to be able to determine what's the area of each of these dimensions, right? And so for this guy, it'd be pretty easy, right? Because you could just trace it or use a circle tool. But our images, our lesioned areas, are never nice little circles. They're always a little bit crazy like this. So you could trace this by hand, and that would work on this image because there's only 3 areas. But on our real images, there will be 15, 20, 100 different little areas, and so doing that is a little impractical. Fortunately, ImageJ and the writers thereof foresaw this, and they include a very nice little plugin called Analyze Particles. And so if you click on the Analyze Particle measure, it pulls up a nice little thing here. It's another little menu. So several things to keep in mind is there's a very cool tool called the ROI Manager. So ROI is Region of Interest Manager. So we want to make sure that all of our data, all of our numbers get sent there, because that's going to be a very important way to sort of manage and assess that. Whether we want to clear results depends kind of where we are in the stage. So at this very early stage where we're just kind of assessing things, we'll generally want to just continue to replace the results. If you want to just add on to the results, as we'll do later, we'll have to uncheck that box. Generally we include holes because if there's a hole within a lesion, it's probably a lesioned area, so we want to do that. And then we can leave the show with nothing. Okay, so now what we're going to do is we're going to input our thresholded image into this algorithm, and let's see what happens. Okay, so here's that ROI manager I was talking about, and you can see it very nicely produced a very long list of all sorts of little spots. So you can see number 1 here was one of our lesions, and number 16 is another one of our lesions, and number 46 is a third of our lesion. But we got all these other little spots. Now that's going to be a problem, right? Because we want to understand how many of these lesions are, and these aren't lesions, these are just noise in the background. So how can we deal with that? Well, it turns out that there's another little input to our ImageJ, or our analysis here, and that is the pixel size threshold. So you can see here it's set to 0, so any size at all, to infinity, or as big as it gets. So this is something that we can adjust. And so what I'm going to do is I'm going to set a lower level threshold. Now in our for the myocytes, the practical example is about 1,000. So if you put 1,000 pixels in here, it's a relatively, it's not a small myocyte sized area. Of course you can adjust this if you find that it's automatically excluding areas that you wish it would include, and we can talk a little bit about how to sort of manage that. But at the end of the day, this 1,000 seems to work pretty well. Alright, so we're going to just rerun this analysis and see how that works. Alright, so now you can see the ROI Manager gives us 3 areas, 1, 2, 3, and corresponding to our larger areas, lesions, which we thought were going to be more important in our analysis. And so you can highlight these guys, and you can say, so 1, so now you see it's kind of yellow. 2, 3, you can do various things with them. Okay, so that's good. So now we've been able to identify these other areas, but now we want to identify the entire background area. So now we're going to go back to our threshold, and we're going to increase that some more until we get the background now being completely black. Okay, so now one thing to note is that we have this area selected. So now if you have any area selected and you go and you run your analyze particle analysis, it's only going to analyze the stuff inside here. So you will only get one image. So if you want to undo that, edit, selection, select none, or shift command A, and that will remove any selections that you have. So now we can run the analyze particles again. Now in this case we want to remove this clear results, because that would of course clear these results, and we don't want that. We want to keep this, but we want to add the next one to it. So what we're going to do is we're going to run this again. We're going to add the results. So it keeps 1, 2, 3, which are our areas of interest, but it also adds a fourth, which is the big area around the side here. Okay, so that's a nice little thing. So the ROI manager we mentioned earlier has some nice little features. One of those is that you can change how the different ROIs are, how they show up. So you can do that using properties. And let's say we want, instead of this to be this nice blue color, we want the outline to be green. So we type in green, and okay, and so now you can see that it's green. So this is all very nice. So now we have our areas outlined, but now we need to measure them. So now we're going to go back to our original image. So in this case, I can just reset our threshold here. And so you can see now that our areas are all highlighted, and our ROI has been, our regions of interest have been applied to the original image. And so what we do is then we highlight all of these, and we click measure. And so now measure will pull up a results table, and you can see various things that are helpful to us. So we have 1, which is the area of lesion 1. It also has mean, which is interesting. So you can see that its mean intensity is about 159. So out here would be about 256, very bright. In here you see regions 2 and 3, they're essentially black, so their areas are 0. So again, we're not going to use this mean number for anything. It's just to help you understand the thresholding idea a little bit. But these areas are what we're mostly interested in. And then you can see number 4 here is the entire area. It's obviously a much larger number. And so then this data is exported. We will save it, and we'll show you how that's done in when we do the actual image. And that's sort of all of the theory behind it. So I think we can sort of move on and start looking at a real image. Okay, so it's closing this. Do you really want to do this without saving it? And then so we don't need to save that data. So it's all good. All right, so now to pull up a real image. So this image here is actually has 2 things stained on it. So it has the IgG is in red. So this is evidence of injury within the myocyte. So these myocytes have lost their membrane permeability, and endogenous IgG has been able to infiltrate inside that cell. We've also included on this particular section a co-staining with laminin. So laminin is an extracellular matrix protein. It defines the outer aspect of the cells quite nicely. So you can see this gives you an idea of what these myocytes look like. And you can appreciate that when a myocyte is injured, that they're surrounded by this IgG. One thing to keep in note is that there's a wide variability in the sizes and shapes, and so it's a little bit challenging at times to make decisions, and it's something that will come with experience. As you look at more images, you'll get a better idea of what a myocyte looks like. You see these small little flecks that look like might be myocytes. Some of these other little dots are probably not myocytes, so there will be some there's a little bit of a learning curve in deciding what is a myocyte, what is not a myocyte. But the important aspect is that everything we decide will be recorded, so we'll know we recorded this as a myocyte. So if we come back, we can always go through and say, you know what, this probably wasn't a myocyte, or these types of things. Alright, so this is sort of a demonstration purpose. Most of our images will only have the IgG, so they will look more like this, right? So this is just the red channel from this image. So I'm going to close that up. Okay, so this is the original IgG that came off the scope. And you can see there's lots of areas of lesions that we want to quantify, and there's some area of myocyte, and there's some parts of this image that we would want to exclude, right? Because this is not myocyte material at all. And maybe even in here a little bit. All right, so let's get started. Alright, so the first thing we want to do when we have this image is we want to make some copies of it, because we don't want to actually alter the original image at all. So we're just going to make a bunch of copies of it so we don't have to worry about accidentally overwriting anything. Okay, so we've got our original. I'm just going to dump that back down there. Okay, so now we have this copy of this image that we're going to use. So we're going to do exactly the same thing, well, a similar thing that we did with the practice image, but one thing you'll notice that this is red. So what we want to do is the first thing we want to do is to adjust the type, and what we do is we adjust it to an 8-bit image, which will turn it into a nice little grayscale. And then another thing that's helpful is because the threshold, or the analyzed particle, recognizes dark on white, we want to flip this image over, so we want to invert it. And so that again can be done, where is it? There it is, Edit, Shift, Command I. We'll invert it and turn it into something that looks a little like this. Alright, and so now you can appreciate sort of these hazy gray areas are what we are interested in tracing, in these darker areas over here. So we can go to our threshold again, and we can start to define these areas a little bit better. So we can move this thresholding around, kind of try to appreciate where, to what extent we want to have this done. All right, so you can see we're starting to appreciate the differences here. So you've got some areas that are black and some areas that are white, and so most of the areas that were of interest are now mostly dark. Okay, so then what we can do is we can take this and we can go to our analyze particles, take this guy, and we can clear the results this first few times, and we're going to run it. Okay, and so now you can see that a lot of our areas got included, but it sort of ate into some of them. Now what would be helpful is to be able to take this image and look at it with these regions on one of these images. And so fortunately enough, there's a way to do that. So if you go to Image, Overlay Table from ROI Manager. Now you'll notice I selected this other image, and so it will take the image that's on the top here and it will add the areas of interest. And so we kind of look through here and we can look at this image and we can sort of compare it to this image. And so let's start on this far edge over here and look at this area here. So you can see it made some indents into this image that probably shouldn't really be there, right? Because this lesion is probably just one big single lesion. But it's drawn some little areas into that lesion that it thinks are different based on the thresholding. So we need to sort of trick the algorithm into thinking that this is all one big area, because it is, right? And so what we have to do is we go back to our thresholded image here, and we need to sort of trick it into thinking that this area is all one big black blob and that it doesn't have to come through here. And so the way we do that is with this little paintbrush tool. And so the paintbrush tool, if you double click, you can choose the pixel size. If you go over to Image, Color Pickup, or Command Shift K, you can get the color picker, which is a helpful little tool. We're just going to use the black and white here. So we're going to do black first. And so what we want to do is we want to sort of cut off this little region here. So we're going to make that a little black bar. We're going to do the same thing here. Draw this down so that it will then recognize all of this area as being a single lesion. And we can continue to reference back, and we can say, you know what, this probably needs to go as well. So we can just draw across here to sort of blacken that in. One thing you'll notice that it took this area off the center, but when we look at our regular image, it's probably right in the middle of the lesion. So I'd be very tempted to just include that, so basically drawing along the side. So now it will recognize this image all as one. So if we take this image and we then rerun our analyze particles, okay, so now you can see this is all one pretty big image, pretty big area, number 13 here. And so if we want to see that here, what we need to do is we need to go to Image, Overlay, and from ROI Manager, which then updates that. And so now you can appreciate how it's included all of this. Okay, so that's good. And then we've got to do, looks like something up here similar. This should all be one kind of biggish lesion. When we look at this, you can see it's sort of cut in various spots. We can include these little spots in within the lesion area. So yeah, this should probably be included. So what I'm going to do is I'm just going to come back in through here. I'm going to draw a line through there to make sure that area gets included. We decided that this was going to be included too. So that's good. These are probably different and appropriate. So I don't know. Let's see about this one here. I'll go to this here. Look at that. I'd probably say that this guy here should be included in the lesion. But I'm okay with this guy not, because it does look like it kind of works down, and that could be a myocyte in there that's holding out and isn't damaged. So then you've got this area here. That looks good. Yeah, so I'd say that's reasonable, right? And so if we want to sort of see how we're doing, we go back to Analyze Particles. Okay, come here, Overlay from ROI Manager. Okay, so that's looking pretty good. All right, so now let's continue on. One thing you'll notice, you get this very bright dot. Now this very bright dot, it doesn't look like a myocyte. Myocytes are not circular, so this might be this is likely an artifact, and we don't really want to include it. So we want to make that image be less to be ignored by the thresholding system. So the way we do that is we switch from our black brush to our white brush, and there we go. Alright, so now we switch to our white brush, and we can come through here and we can just kind of color that away. Okay, and so now that is gone. So then when we analyze our particles again, it disappears just like magic, right? And so when we update our overlay table here, you can see it now ignores that little bit. Okay, so now there's a little bit of other little cleaning up that we have to do. So this area here, I would not be inclined to call that a myocyte. I think that's probably just a random fleck of that just set off the thresholder. And similarly here, this is another little area that's not a positive myocyte. So we need to go in and make sure those areas get excluded. So again, we're using the same little white brush to sort of take those areas and remove them from the analysis. So let's see, there's probably this area over here, doesn't need to be there. Okay, alright, so now let's see how we're doing. So you see I go back between the labeled image and the unlabeled image, trying to assess where this where the injury is and where it's not. So one area that's particularly troublesome is down here, right along the edge. Is you'll often get a very positive area, brightly staining immunoglobulin area right along the endocardial or epicardial surface. Now these are not indicative of damage, so we don't want to include them in our analyses, but they are they do come back positive. So again, we're going to have to use our little tool to get rid of this whole area here and some of this area here, but not all of it, because you can see that there's some myocytes that are positive in here, although it may have expanded this area a little more than it ought to have. And this area up here, if we look at this image, they're not positive myocytes in that area, so we can likely get rid of those guys too. Alright, so what we're going to do is we're going to go to our image here, and I'm actually going to increase the size of my brush. I'm going to make it 50 or so, so that I can more effectively remove these areas, without taking forever to do it. Okay, so that's one area there. So now looking over here, we have to sort of assess what are we gonna count. I'm going to basically remove all of this. I think none of that is positive myocytes. So starting here, working our way over this way. Okay, so that's that. Now this area gets a little tricky because you can see there's clearly a positive myocyte here, but this is probably all just noise. So we have to be a little bit selective in how we do this. It's a little bit tricky with the big brush. I'm going to go back to our, oops, not that small, to our smaller brush. Okay, and so you can see we can kind of work our way in here, and that looks about right. We'll get an opportunity to sort of assess that in a little bit. So you just basically are removing this area here. Okay. Now you don't have to do it perfect, right? It has to just get below that 1000 pixel threshold. So here's another little tricky area. Is one might be inclined to keep some of this, but this is probably extraneous. So we have to figure out how to do that. So I'm basically using this image as a guide. I'm going to say anything to the right of this little hole is not worth including. Alright, so we'll sort of sketch that away. And then we have sort of this random area up over here. I'm going to say it's something like that. And again, we'll assess this in just a moment. Alright, so then this area along the edge here up to here is probably not worth keeping. There's maybe a little myocyte here, this guy. So it's a little trickier to see, to move some of these images around to be able to appreciate that. So that positive area is right in here, so I'm going to get rid of this guy here, and then all of this down in here. So just sort of, yeah, this is a little bit of a tedious process, but it will allow us to really have a good idea of what these lesions look like, which will be very important for us to understand how they're coming about and what it actually means. Alright, so then this whole area up here again is all just noise. There's no real myocytes there. I mean, there's maybe a little artifact in the tissue. It's hard to say. So what we want to do is we want to get rid of all of that positive area. So again, I'm going to go back to my big brush to be able to remove most of these areas here. Now you can see what's happened now is I accidentally selected the yellow one. So I'm going to deselect all. Start over here. Alright, so working my way up, just removing large areas of noise. Let's see, now I wanted to double check, make sure there's, yeah, so this is a little borderline. I might be tempted to keep a little bit of something at the end there, just because that could be argued as a myocyte. Let me get rid of that little extra shoulder there. Okay. Alright, so let's see where we're at. So let's go back here, analyze images, okay, come back here, overlay from our line. Alright, so you can see we got rid of all that noise, so that's good. We got rid of a lot of this stuff, so that's good. This is much better, we're doing okay here. Gotta decide about that little nubbin right here. So yeah, that's probably not a +5, so that's probably not worth we're spending too much effort on. Okay, so let's get rid of that because it's not positive. And then you see this little, we have that positive area there, but then there's this little add-on, which again we don't think is real. Okay, so let's get rid of that, and there's this little tail coming up here, so we'll get rid of that. Okay, so this area I think is in pretty good shape. Now let's assess this other area over here. Alright, so what we have is it's basically including this entire area, which seems appropriate, although this may be a little bit of a call. I might be tempted to disclude all of this stuff over here. And so that's this nonsense here. So I'm going to just come in and on our mask image here, just get rid of this stuff. Alright, let's see how that does, what that does for us. Okay, come back here, overlay from our, okay. Alright, so you can see we sort of scaled that back a little bit, which I think is appropriate. We sort of stop at this area here. We might come up here and say, you know what, this area on the top here is probably not positive. So we can get rid of this stuff coming down to say there. Okay. And then let's see. Whoops. Wrong image. Alright, so here we go. So this looks all good. Matching it up over here. Comes down. We got an area here. You know, I might be tempted to sort of drag this off. Cut those, separate those. I'm going to go back to my smaller brush to do that though. Okay. So again, I'm going to basically draw a line through here saying, you know what, there's this negative region right in this area here, probably coming down a little bit like this. Something like that. Okay, so that's good. So I think we'll keep this stuff heading up that way because that all seems to be appropriately positive. There's this little area of noise out here, which is clearly not on my side. Just a little contiguous thing. We can get rid of that. We kind of move along here looking at the different things. This little shoulder here is probably not positive. Get rid of that. Let's see, we got these guys out here. Decide what we want to do with those. I'm going to say that this area is positive, but these guys here are negative. So I'm going to delete these. Takes a little while with a smaller brush, but again, you don't have to be perfect. You just have to get it below 1,000 pixels. Okay, so keep that area here, keep this here. Now this looks like it might just be edge, and this clearly is edge. So I'm going to get rid of these areas here, and this guy here. Okay, so that's good. And then down here, I'm going to increase my brush size again because it's getting a little more difficult. Okay, so let's do that. Let's get rid of this here to there. Keep this. Keep some of that. Alright, we got this going on. Let's see how we're doing. Analyze. Okay, come back to this image. Okay. Alright, so this looks pretty good out here. When we look over at the lesions, the unlabeled lesions, we can kind of appreciate that. There's this little stuff over here. We've got to decide. I'm going to say that's negative. So this is negative, and that's great. Okay, so then I got to erase this guy here, which we decided was going to be negative. Kind of clean up the edges a little bit. These little outcroppings are rarely positive. Okay, so I think now let's just do one last run through here, see how we're doing. Okay. Alright, so I think we're pretty well done identifying the individual areas. So now if we go to our ROI manager, you can see we have a relatively long list of looks like there's 20 regions in this particular image. Okay, so now what we want to do is we want to quantify the background image. So I'm going to just go and get another image here. I'm going to turn it, do basically the same thing, turn it into 8-bit, invert it, Command-Shift-I, and then we're going to threshold it, which is Command-Shift-T. Alright, and now in this case what we want to do is we want to basically turn this threshold up quite high so that we can appreciate most of the background that is my side. So there's a little bit of red, you can appreciate that just in these images, and that's what we're going to use to be able to identify the specific areas. So we can take this image and we can so if we analyze particles on this now, it's going to identify a lot of different areas, little nooks and crannies. Oh, we might as well do it. Let's just see what happens. Okay, now I don't want to clear results, so I'm going to unclear our results, and I'm going to say OK. And so what you'll see is that you have a lot of sort of big areas, and we've added a handful of of really just one big area, right? 21, which is I guess 20. 21 is just this small little nubbin over here, right? Which isn't really a positive thing, but it's clearly outside the muscle. So what we can do is we can just delete that in the ROI, just so don't pay attention to it. Let's look at our 21. It looks pretty good. It comes in here a little bit and captures some of our excludes some of the muscle that we think it should include. So that's a little area of contention. So we need to deal with that. So I'm going to basically you go back to our brush. I'm going to pull up our color picker again. Okay, I'm going to go back to the black brush because I want to basically seal these off so that it doesn't think that there's anything to exclude up there, because the muscle goes all the way up to the top there. Okay, and then just to make life a little easier, I'm going to exclude these guys down here. All right, so then let's go here. I'm going to take this number 20. I'm going to delete it because we're about to replace it. I select this image, analyze particles, run it again. Okay, and hopefully we just have one more. So now it's labeled 22, but you can see it labels the entire outside of the cell. So that's good. So now let's make that stand out a little bit. So instead of cyan, we'll make it green. Okay. Oops, all lowercase, I guess, is important. There we go. All right, so now we've got this nice little green outline that describes the outside of the cell. And so we come back to our original image here, and we can add the overlay, and now you can see the green is now selected. Now one thing you'll notice is that there's an area in here that's sort of a hole in the tissue that we might be inclined to exclude as part of counting the total muscle area. And so in order to do that, we have to identify another ROI. So it's a little tricky to do automatically. Generally, there are very few of these areas, so it's often easier just to do freehand on these one or two little sections. And so the freehand tool is this guy right here. And so what you can do is you can highlight, oops, you have to unhighlight everything first. Okay, so now we can sort of trace a little area here that is going to be excluded from the total. So you can double click. So now we've got this area. So then what we want to do is we want to add this area. Oh, we do it over here. In the ROI manager, we want to add this last area. So we're going to do that, and it should add an area 1164, right? And so I'm just going to change the properties of this so we can see it, and I'm going to, instead of green, I'm going to make it red. Okay. And so now you can see it shows up red, so we can see how that red's probably not a good choice. Let's try blue. Red might be a little difficult to see on this image because there's so much other red. All right, so from the ROI, you say, hey, you know, that's not too bad. We could maybe expand that a little bit, and so you kind of go back and forth here, trying to make this as best you can. You don't have to kill yourself trying to make it spectacular, because you know these little pixels, you know, a pixel here or there is probably not going to make a big difference. It's all about the ensemble, how it turns out at the end. Properties blue, okay, let's see if that's any better. Okay, you know what, I'm going to call that good enough, because we could spend all day trying to maximize that. All right, so now it's time to sort of document the work and to save it. On the server where these images are stored, every mouse has been given a specific identification table, and then every image from that mouse has its own little directory. And as you start, each directory has a single TIFF file sitting in it, which would be the equivalent of this this original image. Now we've gone through all this effort to identify these particular regions of interest, so we want to be sure that that work is able to be saved and accessed at a later date. So there's several things that we have to do. One is we have to take our original ROIs and go back to one of our original unadulterated images that we want to apply from the ROI our regions of interest, right? And then we want to take these guys and we want to measure them. Alright, so now you can see we have 21 areas of differing areas, which are particularly what we're interested in. This bottom one is sort of the outside total, and then this is one we're going to exclude from the total. And so we'll have to make a document of how that works in just a moment, and we'll discuss that. Alright, so we're going to take this and we're going to save it, so Command S, which will save this somewhere. Here it comes. Wherever we wish. So this is sort of in a little area here where we're doing some IgG quantification. So I'm just going to save it up here. We're going to save, and it'll save it as results. So what we really want to do is we want to enter in up here the number of the mouse. So I don't remember. This one didn't have a number because it's just a... So the number and then the image, something like that, and then the results, okay? And so that, with that, that'll be great. So we can save that. Alright, and then so that saves the data, which of course is important. The next thing we want to save is the regions of interest. And so you go to the More here, and you click Save. And in the same spot, it'll create an ROI site. So here we're going to use basically the same, make sure I use the same number, identification, so the mouse, and then the image number, and the ROI set, and save. Okay, and then the third thing we want to save is this final mask image that we did. Now if we just save this, it'll create a TIFF that image J can read just fine, but nothing else can read it. So we want to create a file that will be much more readable by a variety of different programs. So the first thing we have to do is we take this and we need to flatten the image. So we'll basically merge all of these different layers into a single image. So we go to Image, Overlay, Flatten, and we end up here with this overlay image. Oh, one thing we forgot to do though, so let's go back, is we want to do this, Show All, so that we can see the individual numbers, which is helpful later on when we need, if we're referencing the image from this. Alright, so let's redo that, Overlay, Flatten. Okay, so now we have this image that's quite nicely done. Okay, and then what we're going to do is we're going to do a Save As TIFF, and we're going to call this that, and then we're going to call this overlay. Okay, and then the last one is this file, which we worked on relatively for a while, right? This is where we removed all those areas. We want to save this file too, and we're going to save this again as a TIFF. And instead of overlay, we're going to save this as mask. Alright, so now if we go back to our Finder here, we can, let's see, there it is. We can see now that we've added the mask image, the overlay image. This is our ROI set, and then the results. So now we needed to annotate these results a little bit, so I'm going to open this up in Excel. And so now there was this one here was basically the total area, and so we want to call it such. So we're going to label this total, and this is that area that we wanted to exclude, and so we're going to label that exclude, and hopefully we spell it right. Okay. Alright, so saving that as such will then allow us to and to sort of automate the analysis later. So we will then have a program that runs through, collects these images, recognizes the numbers as lesioned areas, the total as the total, and the exclude is going to be the areas we were trying to get rid of. So anything that we need to include is going to be the total. So the total area is going to be the sum of things labeled total subtracting the things that are labeled exclude. And then the individual areas, the percentages, we'll look at the sum of these percentages divided by the sum of the total area, which will give us the result for that particular image, which we can, after we look at all of the images from a mouse, we'll get an ensemble average for that particular mouse. Okay, so then you want to save this, and you just continue. You can see that it updated quite nicely. All right, so that's the gist of it. So there's not a lot more to worry about. So, all right, thank you very much.